This is the highest the river will be this season, so there's this giant flush of particulate and nutrients that flows through the rivers.
In the early season, May or late April, the Alaskan Arctic is completely snow covered, and with 24 hours of daylight and getting some warmer temperatures, thaw happens.
There's two rivers that we're mainly focused on, the Sagavanirtok River, or we call it the Sag, and the Kuparuk River, which is just probably 40 kilometers west of here.
You see it as first this kind of yellow trickle in the snow, and the color yellow is from all that soil organic matter that's being washed downstream as the ground is thawing and the snow is moving over land. And then basically the whole landscape just turns yellow. That's all that beautiful organic carbon that I love. Late May and June tend to be the sunniest days, so we don't have a lot of cloud cover or rain or snow. And so this pulse of all this organic carbon is coming out of the ground at just the right time when we have a lot of sun.
What we are especially interested in is studying how sunlight affects the carbon that's flowing in the rivers such as this one. There's a tremendous store of carbon in Arctic permafrost, nearly twice as much as there is in the atmosphere right now. So all of this carbon we're seeing melt from the permafrost and end up in rivers and lakes. And what's going to happen to that carbon is a really important question on a global scale.
Most of the waters are shallow. So in the early season, we've got sunlight, we've got shallow water, we've got all this organic carbon coming in, and the water's moving fast, so it's mixing all that up, and it's just continually getting exposed to sunlight. And those are factors that contribute to these high rates of this photochemical or sunlight-driven reaction that turns that yellow organic carbon into carbon dioxide.
If this carbon, which now is in an organic form, in a dissolved form, gets converted into carbon dioxide, it's going to end up in the atmosphere where it can affect all sorts of things, namely reinforcing climate warming and additional melting of permafrost in the Arctic. So it's a reinforcing process.
The conventional understanding is that bacteria or biological reactions are mainly responsible for turning all that organic carbon into carbon dioxide. But what we discovered is that in the Arctic, the waters are cold and they don't have a lot of nutrients, and so the bacteria are very slow, and the sun doesn't care how cold it is as long as it's sunny out and sun is shining on the lakes and streams. That organic carbon will be turned into carbon dioxide. So relative to other places on Earth, that photochemical reaction is actually more important in the Arctic as a source of CO2 to the atmosphere, which was surprising to some.
Whoa, that's a big one.
That's ice.
That's ice.
Yep, that's cool.
Yeah, and the color is dissolved organic carbon.
I'm Rose Corey. I'm a professor of aquatic geochemistry at the University of Michigan. I study naturally occurring organic matter or organic carbon in water. So that means in lakes, streams, rivers. It's the naturally occurring carbon that's made from decaying plants and organisms in water. It's what gives water its brown to yellow color.
My name is Dr. Sarah Godsey, and I'm an assistant professor at Idaho State University. I focus on work in polar and mountain regions. Specifically, I look at catchment hydrology, so I care about watersheds and how water moves through the landscape, how it changes in terms of the amount of water, the quality of water, and the timing, and especially how that timing can influence water quality and water availability.
I'm Tamara Harms. I'm from the University of Alaska Fairbanks. I'm in the Institute of Arctic Biology and the Department of Biology and Wildlife. I'm an ecosystem ecologist and a biogeochemist, and I'm interested in elemental cycles, particularly the bioavailable elements carbon, nitrogen, and phosphorus. These are elements that make up living biomass, And all organisms are trying to acquire these elements for energy and to build their biomass. And so we're interested in how those elements move around the landscape, and water is one vector for moving those elements around.
Water tracks are extremely common in the foothills region of the Brooks Range. Of course, because they're on hill slopes, you need a hill slope for there to be a water track. You don't have water tracks out on the coastal plain area.
Water tracks, which are kind of different than anything you'd see outside of the Arctic region. They're unchannelized. So unlike a stream, which has, clear banks and a bed, these are areas on hill slopes that are more or less linear regions where most of the water flows below the surface.
They don't form a channel that transports very much sediment or cobbles, but they do drain the hill slope of water.
My name is Caitlin Rushlow. I'm a PhD student right now at Idaho State University, and I'm working with Dr. Sarah Godsey and Dr. Tamara Harms on the Water Tracks project. We're looking at the coupled hydrology and biogeochemistry of water tracks, and I'm specifically working on the physical hydrology side of the project. Yeah, okay.
One of the challenges of our work is trying to understand how much water there is, how nutrients are transformed in those water tracks. And really one of the reasons that we care about this is that we don't have these features elsewhere. They're really related to permafrost, so they rely on there being frozen soil.
Anywhere that there's permafrost, that influences where and how water moves through catchments. Water doesn't move through frozen soils and it flows over the permafrost. Places that lack permafrost or have discontinuous permafrost have areas where water can leach more deeply into the soils and then move more slowly through and out of catchments. Where permafrost is present, and especially in the Arctic, the depths of soils that water can move through are very shallow. And so water's interacting mostly with organic soil.
All we're doing today is measuring discharge, water discharge. To do that, we use an instrument that measures water conductivity. And so we'll put it in the water downstream and measure what the background conductivity is. And then we'll dump a salt slug in upstream. So the water itself will mix it by the time it gets to the probe down our reach.
So we're measuring the flow of water using dilution of salt. So I know exactly how much salt is just table salt that I added. And then that conductivity value is representative of the amount of salt in the stream. And so we're just watching for dilution of that salt. And the amount of dilution is due to how much water is in the stream.
It will, go up and down, and then we integrate under the curve, and that gives us our discharge. So that's it. It's really simple. It's a good method for small streams where instead of using water velocity meter, because it's so shallow, you can just dump a little bit of salt and it doesn't hurt anything because it's such a small amount and it just flushes right out.
We have a full year of data and samples analyzed this current field season that we're in, all of those bottles of water in the freezer, and Caitlin is working through calculations from her loggers. And so about this winter, we'll start to have some good answers. We have some observational answers. We know how quickly they respond to precipitation, for example. We know that the hydraulic head suggests that water is moving from the hill slope to the water track. We know that huge fluxes of solutes happen during snowmelt, and then elements become very scarce later on during the year. And so they're transporting a lot of material during snowmelt, and then those elements become very low concentrations this time of year.
We have many hypotheses about water tracks, and there's not a lot that's known about them. So there are many questions we can answer, I think, and that's really the point here, is to understand more about the Arctic, specifically water and nutrient use in the Arctic. Streams are fairly well understood, but the link between the terrestrial and the aquatic environment is less well understood.
I had tremendous mentors, so I was lucky to just find faculty who are really excited about mentoring undergraduates. And they encouraged me and they spent a tremendous amount of time with me. And then when I went on to graduate school, I had, again, tremendous advisors and committee members who teach you ways of thinking about science and ways of looking at the world. And I think without that, I wouldn't have continued on. Half of the fun of this gig to me is to be able to work with these sharp young students who are just finding out about these fields for the first time. Some of them are a little bit more experienced, but they come at it from very different perspectives. And so there's a chance to help shape them into scientists, but at the same time let them bring their background and expertise to bear on the project.
My name is Rebecca Risser and I'm a master's student at the University of Alaska Fairbanks. In the Arctic, the streams here are extremely nutrient limited, and most of the nutrients they receive are inputs from the hill slope. We want to know something about where those nutrients are coming from, what makes them move there, and through that we'll know something a little bit more about what's going on in streams and what potentially could happen in the future. They have long-term phosphorus additions on the Kuparuk River, which is the basin we work in, and they see things like an increased size in the young of the year Arctic grayling beneath their phosphorus dripper, and huge changes in the biotic community there, more algae, all the consequences you would expect from eutrophication in streams in the lower 48 and other areas where you have additions of large amounts of nutrients and just grows to take over a pond or a stream. And further than that, the Arctic is one of the most land-influenced oceans on the planet, so what's traveling into it from the streams has a huge effect on Arctic fisheries and aquatic life. So you can see plumes from Arctic streams or Arctic rivers out dozens and dozens to 100 miles from the coast.
We don't know yet how much of that permafrost carbon will go up to the atmosphere as carbon dioxide or be exported to the Arctic Ocean and basically stay there for a longer amount of time.
It starts off with you take a sample from a thermokarst impacted water or a surface water. We actually bring the sample back to lab, we measure all the different nutrients, and we characterize the carbon that is in the solution.
Set up these large experiments on the tundra where we'd expose it to the natural sunlight over the course of a day and basically measure the amount of CO2 produced.
Right now we're just labeling sample bottles, and each bottle will be filled with filtered water. They each get a different color of tape because each bottle in these lines will be analyzed for a different component or analyte in the water. Most of them just end up getting shipped home to our labs in Michigan, where we'll run them on machines down there because we don't have the equipment to do it up here.
And we collect, I don't know, 1,500 samples over the course of a season.
In our experiments, what we do is we take water, like from the Kuparuk River or somewhere, we filter it, and we take that and put it out in the sun and just let it be photo exposed, or let it soak up sunshine, essentially.
Initially, when it comes out of the soil, it's really good at being turned into carbon dioxide by sunlight. And we think that's just because this is carbon that's never seen sunlight before.
And we also then take the same water and keep it in the same exact treatment, but then we wrap it in aluminum foil and leave it out there right next to it, so it's kept dark, completely dark.
There's all these what we call labile molecules, or when it's labile, that means that the reaction is fast. You know, this is stuff that can be easily converted into carbon dioxide.
We take that water in and we essentially feed it to bacteria and look at what they do with it and how active they are given these two types of carbon, this light carbon and this carbon that was kept dark. So what I've measured was bacterial production, which is a snapshot of just how active those microbes are.
The difference isn't in the process of the bacterial production, the difference is in the carbon that is used to feed the bacteria.
Right. Mm-hmm.
So the sunlight changes the carbon and makes it more readily used by the bacteria, whereas compared to the carbon that was not exposed to sunlight. So that's the difference between light and dark.
You can think of it like how well the food is baked for these microbes. So if you take a frozen pizza out of the freezer and just give it to these microbes, they don't like it. But if you take it and you put it out in the sun and bake it for a little while, they'll eat it a lot faster and turn it into CO2 a lot faster.
What we don't know is if you take carbon coming out of the soil, how much of that is the easily converted carbon dioxide? 50% of the organic carbon in the soil? Is it 10%? And that's something that we're trying to get at with our experiments.
One observation we've made is that we saw that the soil is almost like a microbial seed bank, and that there's a whole bunch of microbes just hanging out in the soil waiting for the right conditions where they can really flourish. And maybe they get washed in after a storm, they find themselves in a stream and say, hey, this is where we like to live, and they start gobbling up everything in that water and dividing and multiplying.
We'd collect that water, set up these large experiments on the tundra where we'd expose it to the natural sunlight over the course of a day and basically measure the amount of CO2 produced. And the control for those experiments were just water kept in the dark.
And you can see that the larger volume samples are in chocolate amber vials, and then the glass vial samples are wrapped in aluminum foil.
And then water fed to bacteria under a variety of conditions. So we could basically pit bacteria against sunlight and see how fast they could each turn that organic carbon into carbon dioxide.
Certain microbes flourish under the two different conditions, and that tells you something about the food that you fed them, how the light changed their food source and allowed different types of microbes to really benefit and grow and multiply. The type of dissolved carbon in the water plays a big role in deciding which microbes grow the best.
Looking at that over landscape had suggested that sunlight was important, but we hadn't measured the actual rates. I didn't expect it to dominate or be so much higher than bacterial respiration.
It looks kind of like chocolate milk in a lot of ways, and you can just watch that billowing out into the lake as it mixes with the much clearer lake water. To collect a sample from a permafrost thaw slump such as this, all of this water and ice melts and collects a bunch of the mineral and sediment particulates, and it travels down and it kind of goes out into this mud flat, which is really difficult and treacherous to walk on. Oh boy. Careful! Careful. Yeah, it's really, really treacherous, and it's going to slowly make its way to a little tiny dribbling stream into the lake. So when I collect a sample, I first go to that little stream, which is usually just a trickle. So I have to make a trench to collect enough water from that trickle. I can put in a pH and conductivity probe, measure the temperature, so get some of the physical characteristics that's in this water. And then I have a 8-liter bottle, which I have to first rinse out just to make sure we don't have any residue from anything else. So I pour that in, rinse it, and dump it out. And then when I'm ready to collect, I use a smaller bottle to just collect water and pour it into the larger one. And that's the water we'll actually take home and analyze further in the lab. When you have a thaw slump like this, what happens is somehow permafrost gets exposed to sun and to heat during the summer, and it melts, slumps away, and thus creates a sort of a chain reaction where as it slumps away, it exposes more frozen material that's never seen sunlight or warmth for the last several thousand years, and then that melts away too. And what happens is you get a huge slump forming where more and more material just keeps falling away and falling away. If you can see around here, it can be really a huge feature and can have a drastic effect on whatever is downstream of this material. In addition to that, I also walked over to collect another inlet to the lakes. It's not impacted by a thermokarst failure. It's a clear little stream. It's a deep enough stream that I can just submerge my bottle. Then I fill it with about 8 liters of sample water, again, to take home and filter. So we're really interested in what's happening to the lake as this giant thaw slump continues to grow. There's a very, very huge amount of organic matter that's just been frozen in this permafrost for thousands and thousands of years. And what we see happening in thaw slumps such as this, that carbon, it melts out of the soil and gets released, and then usually ends up in some sort of aquatic system like a river or a lake. So to understand that, we've got to know everything that's coming into the lake and what's happening while it's in the lake and what happens when it leaves the lake. And then how it may affect the lakes that are downstream. This lake flows into another, which flows into another, which eventually flows into the Oxarkoye Creek, which will flow out to the Arctic Ocean. What's really remarkable about these slumps is how quickly they really do grow. In the course of this season alone, this has easily quintupled in size and dumped tons, literally tons of material into this lake behind me. Right now, I'm on the roof of the dry lab at Toolik Field Station, where it's a fairly flat surface that's soaking in a lot of the sun's rays and not getting shaded by many of the things around here. So it's a good spot to leave things out in the sunlight. One of the things we're most interested in up here in the Arctic is how sunlight alters and degrades organic matter in rivers and lakes and in natural systems. To help better understand that, or show how light can have a profound effect on organic compounds, I thought I'd take some less natural mixtures of organic molecules, such as sodas like Coke and Orange Crush, and just expose them to the sun. I've tried to get some more naturally colored things like apple juice, Earl Grey tea, and coffee. I took duplicate samples, one that I would leave out in the sun, and then another that I would wrap in foil and leave in the dark in a styrofoam box next to them, so they're still under the same temperature and weather changes, but the only difference is that these ones are getting lots more sunlight than these ones. It's actually kind of interesting that things that are left out in the sun are effectively sterilized. Like all the microbes that would be in that sample get killed off by the UV light. Not unlike the same UV light that kills our skin cells and gives us a sunburn, it kills all the microbes in these samples. I can show you the dark control samples now. For instance, here's the Mountain Dew that's been left out in the sun and the Mountain Dew that was left wrapped in foil in the dark. One of the most striking, the Coke that's light and Coke that was kept in the dark, and the Orange Crush that was kept dark compared to the Orange Crush that was kept in the light. Some black coffee that was kept dark. Ginger ale, it started pretty light, but it still retained that tan slight color. Apple juice. And the Earl Grey tea. The effects of the sun on organic matter are often overlooked and not really factored in, but it's a major control on what happens to all these compounds as they're released from, in the case of what we saw yesterday, thermokarst slumps, where organic molecules have been frozen for 10,000 years in the dark. Now they're up at the surface getting blasted by UV light, and that's going to totally change the possibilities of what's going to happen to them, whether they're going to be just coast out to the ocean and get buried out there in the sediments or in the deep oceans, or if they're just going to become CO2 through microbial respiration or other carbon processes. Photodegradation is happening all around us. It's just not something people often think much about. And I know I didn't. And it happens pretty quickly, so it's a cool phenomenon.